separation, I have an image of oil and vinegar here as an example of liquid-liquid phase separation, where the oil and the vinegar phases aren't compatible with each other. And so they're both liquid, but they hang out apart from each other. And this is actually really interesting because we're now finding out that the cells use a similar principle to organize themselves. So I'm going to be starting off going over what and why are condensates in liquid phase separation, why do I study them, and how are condensates and liquid-liquid phase separation used to organize the cell. So first of all, when we think of organization of the cell, we tend to think of organelles that perform specific functions, like the mitochondria that's wrapped in a membrane to contain reactive oxygen species or other chemicals that you might not want around the cytoplasm when you're making ATP, or the nucleus, which wraps the DNA in a double membrane that helps for compartmentalization and information transfer. But condensates and liquid phase separation are these ideas where you can have a non-membrane wrapped organelle where you concentrate certain molecules like proteins and RNA that you need to perform a specific function without enclosing it in a membrane, allowing for really dynamic processes. So one example of these is the stress granule. I have here an image of cells where in magenta is a stress granule marker G3BP1, and green is TDP43, an ALS associated protein. And when you add sodium arsenite or stress to cells, you get these puncta. And if you get rid of the stress, it goes back to normal. And these are really remarkable because in these granules, you can concentrate certain molecules like RNA and some proteins, or exclude other molecules like cytoplasmic proteins. So this is a different way that the cell can use to respond to its environment. And in fact, this image is an artistic rendition of a stress granule, where I have here the endoplasmic reticulum membrane, and then the stress granule in the background, and a piece of RNA being unwound, and the stress granule is being disassembled by some protein machinery. So stress granules are one example of these kinds of things, but there's several. So stress granules are involved in RNA storage, where you might want to store some RNAs and proteins during stress so that the RNA isn't translated, and then when the stress passes, you can go back to normal and release these RNAs for the cell to help survive the stress. In the case of neurons, you might want to transport RNA for protein synthesis at an axon terminal. You need to move the RNA all along the axon, and RNA transport granules are phase-separated granules that are helpful for that. Ribosome biogenesis, so making the ribosome machinery, is done in the nucleus, where the ribosomal RNA is made in the nucleolus. So you can also have reaction hubs like in the cell or in the test tube in the case of this paper, where they had sustained enzymatic activity and flow in crowded protein droplets, where they had all these proteins and substrate would go in and leave, and leading to continuous reactions. So another group even poses that they can be used for biological noise buffering. This kind of paints this picture that there's a lot of utility of this kind of organization of the cell, and it's used in a lot of different ways. So this is kind of why we're interested in them. They're very, very important for understanding and regulating life. And I came across them because of my background on amyloid. When I was in the Eisenberg lab, I worked on this protein FUS that forms these liquid droplets. And then from these phase-separated droplets, they'll actually grow amyloid fibers, which is a solid protein fiber out of them. The FUS is famous for being aggregated in ALS and some other neurodegenerative diseases, and this kind of led to the hypothesis that maybe stress cells would form stress granules, and these stress granules look a lot like the beginnings of where we might form amyloid fibers in the cell by concentrating them together. So the original idea to try and interrogate this was when you have in amyloid diseases like Alzheimer's disease, you'll have neuronal death in the brain, and if you zoom in on someone's brain and stain, you might see neurofibrillary tangles of tau and plaques of A beta, And these are protein aggregates that if you zoom in farther with an electron microscope, you can see these amyloid fibers, and then you can do cryo-electron microscopy to get their structure. And the question is then, how do we get these amyloid fibers associated with these diseases? So that's why I study them, to try and understand this process, and then in trying to understand them, I want to know how these condensates are organized. So this is briefly some energetics of liquid-to-liquid phase separation, and the idea is that it's kind of entropically unexpected. So what I mean by that is that entropy is kind of like mixing things. So if you add food coloring to water, it just kind of dissolves over time and spreads out. But when you have two demixed systems, they just stay in the granule. They don't dissolve the whole place. So this is kind of unexpected. The equation that we use to describe chemical reactions, Gibbs free energy, where if your Gibbs free energy is negative, it proceeds forward. And S stands for entropy, and this is roughly kind of like how things want to diffuse over time. It's defined as disorder, so you go from a nice orderly drop to filling the whole glass and becoming homogeneous and disordered over time. Where you just have these particles and it'll slowly bounce around over time, leading to the food color spreading out. So the delta H term is the enthalpy, which might loosely be the attraction of the bonds. And here we can fight diffusion, right? So if your bonds are strong enough, you might not diffuse away. And this is very tied to temperature in the sense that, apologies, so if you multiply the entropy by temperature, so for hot water it diffuses faster, this fills up the glass faster, you multiply by the temperature and this can give you more diffusion more quickly. But in a cold glass there's not as much energy for that entropy term, so it takes longer. So in this case, when you have ice, the bonds, the enthalpy provided by those bonds fights this wanted disorder to fall apart. And so when we're thinking of phase separation, we're kind of playing with this idea of things wanting to fall apart and the bonds to keep them together. There's the classic. So when I was studying FUS, it was remarkable because it formed amyloid fibers, but only when you cool it to like 4 or 5 degrees, where it undergoes phase separation and forms fibers at low temperature, but is soluble at higher temperatures. And this is kind of opposite amyloid fibers of alpha-synuclein, which are found in Parkinson's disease, where they form stable fibers at normal physiological temperatures, but you can actually cold denature and melt these fibers at 4 degrees and below. So amyloid fibers are very much dependent on what kind of protein it is and how this equation gets satisfied. And how people think of liquid phase separation currently, some models are this idea of proposed by Dr. Holthaus, or an introduction to the stickers and spacers framework as applied to biomolecular condensates, where they think of proteins as these long chains, these polymers that have stickers which stick together and spacers in between them. And these stickers, so if you have kind of water or these polymers, we use this Flory-Huggins modeling, where as these polymers dissolve, you can kind of see that they'll contact with each other, and this might be stickers coming together. So as you have a fully mixed state and then the polymers interact with each other through their stickers, you have these interactions which then can pay for the phase separation. So we have this polymer, stickers, and spacers model, but then there's also another way to wonder, as opposed to just the polymers and the protein, what the water is doing. So we consider that in solvent influences. And this is brought up by a couple recent reviews, like in Aqua Veritas, the indispensable yet mostly ignored role of water in phase separation and memberless organelles, or liquid-to-liquid phase separation, Ask the Water. So here are some physical biochemists saying that we should be paying attention to water to understand what's going on with phase separation. So now when I want to understand how the condensates are organized, I want to understand what the polymers are doing and also what the water is doing. And now is a good time to break out this quote that I really like by Dr. Box, a statistician, is that uh, all models are wrong, but some are useful. So reality, this thing here is saying that reality can be so complex that equally valid observations from different perspectives can appear to be contradictory. So we have this weird shape here. This person is saying there's 4 prongs. This person is saying that there's 3. It's not too different than someone saying there's stickers that hold us together, or the water is important. Both are valid. In my book, we just want to understand their contributions. So as I'm going through this talk, I want to be really careful about what I'm saying is data. For example, this weird shape here is data. It just exists, but then the people talking about it and interpreting it make the model. Right? So when we're looking at the people looking at the data, this is us trying to understand what's going on. And then I have this other symbol here that I call overlap, and this is me trying to bridge ideas together. So data is like an experimental outcome, a model is an interpretation of what that means for us, and then the overlap is when I'm coming in and trying to mix ideas together and combine them to synthesize it. So hopefully this can be really helpful for understanding the things that are peer-reviewed that aren't, and how these ideas fit together. So now we want to understand how these polymers and water fit together, and to do that I'm going to ask, when inside the condensate, how do proteins change and how does water change its behavior? So we have the single phase and then we have a phase transition. We can do this in a test tube in addition to a cell, where you could do something like a temperature change and induce a phase separation. So the question is, does the protein change or does the water change? And I argue yes for both. So my model to interpret that comes from data from some papers. So the first one is from the Fawzi group, which looks at HNR&PA2, and they claim that HNR&PA2 undergoes liquid-liquid phase separation and remains disordered in both the single phase and the droplet phase. But when they do NMR on it, they claim that there's heteronuclear NOE values for the phase-separated HNR&PA2 low complexity domain are higher than those of the monomeric reference sample at high temperature. Changes in these values are both consistent with slowed local motions compared to the monomer. Their last bit, however, local motions are more rapid than in folded proteins and correlation times remain in the nanosecond range. So what they're saying here is that changes in these values are consistent with slowed local motions. So the protein is still unfolded, but it's slightly slowed down in the droplet phase, according to their NMR. And then what's going on in the water, to the water inside and outside of the droplet? This is what the Habenith group was asking with looking at FUS by terahertz spectroscopy. So terahertz spectroscopy is going to be shining light onto your sample and measuring its absorbance and dielectric. So water has multiple different ways that it can absorb and move, absorb light, and that corresponds to motion. So it can absorb light that changes its vibrations of its chemical bonds, that changes its ability to rotate, like when you put a cup of coffee into a microwave, it's causing it to rotate, or translation. And so terahertz light seems to be really good at detecting these water motions in the hydration shell of proteins. So they use this technique of terahertz spectroscopy to find that our terahertz fingerprints of the water network inside FUS droplets have now revealed 2 changes. A reduced wrap water population, previously shown to be hydrating hydrophobic patches, and more water molecules with constrained librational motions are simultaneously observed, as expected for tetrahedral water inside the highly concentrated droplets. So what this is telling us is that when the stickers come together, these hydrophobic patches, it gets rid of that hydration, and then the remaining water is more tetrahedrally ordered, and we'll go into what that means later. There's another piece of data that I really want to cite where you don't need proteins to do liquid-liquid phase separation. You can just use 2 different polymers that are kind of incompatible. So one example is PEG and the other is dextran, and these 2 mixed together will form 2 different aqueous phases. And so even without proteins, this is still a phenomenon that we can capture. And this group looked at the infrared spectra of these 2 phases. So they're looking at bond vibration. And they are finding that when they look at this large peak for water around 3,500 to 300 centimeters, that they can decompose this large peak into smaller peaks, and these smaller peaks correspond to different coordinations of water. So this one will be like tetrahedrally coordinated water that they're talking about, where all four donors and acceptors are bound. And then there can be less coordinated water. So they can do, if they observe different peak shapes, they can get different components contributing to that. And what they find is that when they have PEG and sodium sulfate, that compared to just water, this top phase has a different decomposition than the bottom phase. And this tells them that there's a different, for this third Gaussian in particular, that the hydrogen bonding structure is different in the two phases. So between these two so between these data, we can start to put together a loose picture. One of them being inside the droplet, the protein has slower dynamics and lower entropy, as told to us by the NMR, and that we pay for the entropy decrease, the ordering in the droplet phase, with better bonds or stickers. And so this looks like at equilibrium, your delta G is the same, but outside the droplet, you have poorer bonds and you have more disorder, whereas inside the droplet, you have less disorder, but you pay for that by having better bonds. And the water mirrors this, as told to us by the terahertz stuff, where there's more tetrahedral or better bonded water. We can see that outside the water should be more disorder and less well bonded, and inside there should be less disorder and more energy from the bonding. So this kind of builds a general overlap of what's happening inside and what's happening outside the droplets of these molecules. That inside you're a little bit more constrained and you have a little bit better bonding. And that's how you fight the desire to fall apart. So now we have some ideas to hold on to for what the polymers are doing and what the water is doing inside these phases. But to really sell this home, I wanted to understand, how does water actually behave inside and outside the condensate? When I'm saying it has different bonds, what am I actually meaning by this? What am I trying to invoke? So to do that, I want to cover three main points about what does it mean that water behaves differently. The first one is, how does water behave alone? The second one is, what happens, so if I just have an empty glass, how is this water behaving? Then how does water behave when it's touching something? So if I throw molecules in and it's touching water, then how is this hydration water changed? And the last part is, if I want, what happens when water stops touching something? And this means precipitation, when it falls out of the glass. So to start off, I'll go over water behavior alone and citing anomalous properties of water. So water is really common to us, and I didn't think too much about it for a long time, but it's one of the most anomalous chemicals that doesn't fit our typical models that we've come across. I take a lot of this information from this website maintained by Dr. Martin Chaplin at London Southbridge University. Highly recommend checking him out. He's he's curated some 4,500 plus water references over the years and put together some really cool stories. But one of them that he talks about is how water boils at a higher temperature than expected. So if you look at like carbon, silicon, germanium, and if they're all having hydrogens bond to them, that as you get bigger in molecular mass, you get a higher temperature, because it now takes more energy to make the thing with higher mass fly away, right? When we apply the same to water, we see the same kind of trend, but water is sticking out. When you're bound to oxygen, you behave really differently. You have a much higher boiling point than expected. And the origin of this is, so that's the data. The model to understand this is that the electronegativity of oxygen kind of takes the electrons from hydrogen, leaving them a little bit positively charged. So we have this dipole, and this means that water can form hydrogen bonds based on this charge difference. And it has two donor hydrogens and two acceptor oxygen lone pairs, which allows it to make up to four bonds with neighboring molecules, leading to this kind of tetrahedral arrangement, right, if you have all your bonds satisfied. The next anomalous part of water is that water diffuses slower than expected. And this comes from this really, really efficient paper from the 1980s titled Self-Diffusion Coefficient in Hydrogen Sulfide Along the Coexistence Curve. And in my favorite 2-sentence abstract ever, they measure the self-diffusion measurement in liquid hydrogen sulfide along the coexistence curve are reported. The results are compared with similar measurements in water. The anomalous behavior of water is thus made experimentally evident. And how they show this is they look at the self-diffusion time by different temperatures of hydrogen sulfide, and then when they compare this to water, they see that the expected straight line is deviated. At low temperatures, water diffuses much slower than we'd expect. And this would also be explained by the really strong hydrogen bonds holding them together in this regime. So the last water anomaly that I want to talk about is water's anomalous maximum density at 4 degrees Celsius. So what this is inferring is that normal chemicals that aren't anomalous, like benzene or liquid benzene, if you have liquid benzene and liquid benzene ice cubes, the ice cubes will sink. But if you have water and water ice, these cubes float. So this is really anomalous because as you decrease in temperature, you expect to get more and more dense and sink. But from 4 degrees to 0 degrees, water actually increases its volume, which decreases its density, which is why ice floats. So that's really peculiar, and we're trying to understand why that is. And in trying to understand and explain that, I came across this model called the two-state water model. And the two-state water model is not necessarily true, but it's a now common and gaining some traction in explaining the anomalous behaviors of water through this lens where water is composed of two different states that simultaneously exist in the liquid. A low-density form, LDW, where you have optimized hydrogen bonds that expands to create a larger volume, and you have an average around 4 hydrogen bonds per water molecule. On the other end is high-density water, or HDW, where you have bent or broken hydrogen bonds, and this leads to a smaller volume and an average of 3 or 4 hydrogen bonds per water molecule, which is less. So many people have come across, or several different groups seem to have independently come across this theory, one of them being this paper on the existence of two states in liquid water, impact on biological and nanoscopic systems. And here they look at different physical measurements like dielectric constant, and they see that this line has this slight bend in it where it changes. Normally we expect a slight or a straight line, but instead for all these physical properties in water, they keep finding these bends where it kind of breaks around 50 to 60 Celsius. And they interpret that as evidence of two states in liquid water that simultaneously exist. There's other groups like Dr. Wiggins, who talks about life depends on two kinds of water, or water-like anomalies as a function of tetrahedrality. So people have kind of tried to come up with different models to explain this anomalous behavior. And I quite like the two-state model. So how is water different than something that we consider normal? So here I have 2 mercury atoms, and I'm measuring their attraction, their chemical potential to move together. So, and R is how far apart they are. So if they're quite far apart, then there's some attraction until they have optimal bonding or van der Waals interactions when they're touching. And then if they get too close, the electron shells repulse each other and it will move them apart, energy to push them apart. And this kind of creates this ideal liquid, and so if you have this molecule and it's trying to choose who to hang out with, mercury 1 on the left or mercury 2 on the right, then these energies look kind of identical. So it doesn't have much of a preference. But what is unique about water is this double well potential, and this double well potential comes from the hydrogen bond. So in well 1 we have high density water where you have broken hydrogen bonds and fewer average hydrogen bonds per water molecule, and you're now relying on van der Waals forces or interactions And this is disordered and entropically favorable, which is kind of why it's Y. Uh, but the low density water form 2 here has optimized hydrogen bonds. And the optimized hydrogen bond is the most energy you can get from a hydrogen bond is when it's straight. So you have the oxygen in line with the hydrogen in line with the bonded oxygen's free electron pair. And if you start to deviate from that straight line, you don't get the energy. So low density water is really narrow because the water or the hydrogen bonds between these two molecules have to be kind of perfectly in this narrow range. And this actually pushes them apart, which is why it's like farther out now. And leads to this tetrahedral, this want to be arranged in a tetrahedral pattern. And this actually makes the energy a little bit different. So if this water molecule is trying to choose who to hang out with, the high density or the low density, the high density water is entropically favorable. You get more energy from the disorder, but you have poor enthalpy from bonding because of the van der Waals and the poor hydrogen bonds. Whereas low density water is the opposite. You have an entropic penalty because you have to stay nicely aligned, but you do get more energy from the hydrogen bond. So this is the model that kind of supports, or that two-state water works off of to explain these water anomalies. And it talks about bulk water as being a mixture of these two, where when you bulk water would have pockets of low density next to high density water. And because low density water wants to expand to optimize the hydrogen bonds, it pushes out on the other molecules, and this is unfavorable. And so the high density water wants to attack or push back, and this will collapse some parts of low density molecules. So you can actually now kind of move these packets of low density water around by these two phases attacking each other. So this is kind of how this model looks at bulk water. And the main takeaways for me are that low density water is less active and high density water is more chemically active. And what activity means is just how much water is available to bond to something new. So if water is unlikely to make a new bond or make a bond with a new molecule, it's lower in activity. Because like in low density water, all of their bonds are satisfied. And if it's more prone to do that, it's higher activity. So this kind of brings us to an interesting question. Can liquid water have structure? Because I'm talking about this like tetrahedral arrangement. It kind of implies a crystalline structure. And that's what I pose, is that we can interpret liquid water as a distorted crystal structure. And this comes from a 2023 science paper where scientists create a weird new type of ice that's almost exactly as dense as water. And this is truly remarkable because we haven't seen ice with the same density as water. Ice cubes like to float, right? And so how they got to here is that they start off with an ice cube where you have this hexagonal packing of water molecules, and then they put it into a shaker with steel ball bearings and shook it around and broke up the water, or broke up the ice chunks until they kind of made these really, really small chunks of distorted ice crystals that they called medium density amorphous ice. So after they would start off with ice with a low density of like 0.9, and then after 40 ball milling cycles, shaking it 40 times, they would have a powder where they've now achieved a density of around 1. And 1 is really peculiar because that's the density of liquid water. But when we've solved structures of ice, it's either low density or high density, with low density being around 0.9, and then high density being around 1.1 or plus. So when they got medium density of 1.06, this was a big find and had never been observed before. And when they find the structure, it's again somewhere in a mix between the low density and the high density states, indicating kind of a distorted crystal of a mixture of these two. And to be sure, this data and the data that they share of the ball milling cycles exists. The model that I pose of water being a distorted crystal is an interpretation of that, and then me attaching the two-state water theory to this is the overlap of me trying to connect these results to the two-state water theory. So now we have this idea of water as a distorted crystal. So then how does water move through this distorted crystal? And this brings up a jump model, because we normally think of a crystal like a salt crystal where these particles, they won't really move much, but water maintains this kind of crystal but with defects. So what I'm really asking as a consequence of this tetrahedron desire to be tetrahedral. So what I'm asking is, how fast does it take to get from one position of the crystal to an adjacent position? And for water to diffuse one diameter of itself, that takes about 12 nanoseconds, or 12 picoseconds rather, for water to go from one area in the crystal structure to the next area. And this time is everything. So in me talking to you, we might measure this conversation or the words I'm saying in seconds. We might measure like my heartbeat in hundreds of milliseconds or tens of milliseconds. Protein conformations are through microseconds. So if an enzyme or large protein motions would take microseconds, protein tumbling would take 10 to 100 nanoseconds. Side chain fluctuations would be on the order of a nanosecond. And then large water motions take around 10 picoseconds. And hydrogen bond breaking and creation takes around 1 picosecond. So these two times together, for water to move from one node in the crystal to the next is about 12 picoseconds, and the hydrogen bond lifetime is around 1 picosecond. So this tells us that it's changing its hydrogen bonds maybe like 12 times for every second that it jumps to the next area. And this makes it quite a bit different than the ideal fluid, is that it's changing and rotating its bonds much faster than it's moving. And that is really peculiar. And this group in France published this 2006 science paper titled A Molecular Jump Mechanism of Water Reorientation. And what they're saying is this water reorientation mechanism involves large amplitude angular jumps rather than the commonly accepted sequence of small diffusive steps, and therefore calls for a reinterpretation of many experimental data wherein water rotational relaxation is assumed to be diffusive. So what they're saying, as opposed to things moving all around each other very quickly and evenly, that water will just have these large amplitude jumps instead. So if this molecule, or water molecule OA, is trying to diffuse to the right, then the mechanism that they pose that gets there is the first thing that happens to happen for it to go over is that this other water molecule above it, OB, needs to form a hydrogen bond to take this hydrogen bond's place. So this water molecule will rotate up to make a hydrogen bond with the OB, which then should allow for the release of this hydrogen bond that's holding OA back so it can translate or diffuse to the right. And this is the system that they pose. And this is interesting because it requires these water molecules to have very large rotations in order to allow diffusion to exist or to occur. This result was later expanded in this 2023 paper called the Collective Burst Mechanism of Angular Jumps in Liquid Water. So now, as opposed to just looking at one water molecule diffusing, they're looking at groups of them. And what they're trying to do is they find that look at the dipole moment of water. So because it's polar, it has some directionality, and it's going to diffuse over time of femtoseconds in their simulation. And they're going to see that occasionally there will be small angular jumps, but then rarely there will be this really, really large angular jump. And that large angular jump corresponds to allowing that diffusion to take place. And what they find is that when they look at the water molecules that in a certain window have undergone a large diffusional jump, they highlight them boldly here, and you can see that they're clustered in areas. They're not spread out evenly. So that's where they come up with the collective burst mechanism of angular jumps in liquid water, where you get these small collections where all the bonds rotate very large directions at the same time. They expand upon this by looking at the density of water around these jumps. So in the small jumps, which is like around 0 degrees or to 60, the density of water is what's expected, but then around the areas where these large jumps around 60 degrees are occurring, there's lower density. So it kind of infers that at areas where there's low density or distortions in the ice crystal, or the water crystal rather, then that's where these jumps occur. So this they then take this as the water diffusion proceeds via hydrogen bond jump exchange mechanism, and again, this is how they so these large angular jumps are how water moves through this kind of crystalline network. So this kind of sums up the water behavior alone. What does water look like just in a plain glass? The next thing I'm curious about is what happens when water is touching something. So whether you pour salt, protein, or the water that's touching the glass surface, how does that change its behavior? To get at this, I want to start by looking at this idea of a meniscus. So if you just have a test tube where the water is climbing the edges, then if we look in at what the water is doing, the water closest to the edge is kind of immobilized and low activity, and the water in the middle is higher activity. So water wants to move from high activity to low activity, which is why it kind of climbs, or one way to see it is why it's climbing the edge of the glass. And if we were looking at diffusion of water in the middle of the glass, then if we try and look at its path over time, all these water molecules look about the same. But if we look at the edge of the glass where it's hydrogen bonding, that bond's kind of immobile as being part of the glass, so it doesn't diffuse very far over time. So it's paid this entropic cost where the hydrogen bonded water next to it won't diffuse as much over the same amount of time. And then by the time you get to the middle and the bulk, it becomes homogeneous to the same as the others. But this paying of the entropic cost for immobilizing is what gives us this lower density, or this lower activity water that promotes ideal bonding. So water activity is most often used describe kind of how we can dissolve things in water. So if you have an empty glass, you have perfect activity, or activity of 1, and then as you add sugar and the water molecules touching the sugar are coordinated in hydrogen bonding with the sugar, that decreases the activity because these water molecules are no longer available to do other things besides hang out with the sugar. They're no longer as active. Right? So the more sugar you dump in, the less activity, and eventually you get so much sugar where you can't put any more in, and that's when you run out of free water. And this is really useful to us for food storage and spoilage. The reason why we make jam is because when we put in so much sugar, then the fruit is preserved because there's not enough available water for anything to live there, to grow. So I'm going to need to grab my charger for my thing real quick. I'll be right back. Yeah. Let's take a quick 5-minute intermission. Might ask if there's any questions. I'll be right back with my charger. expect that. So okay. So I think I'll keep going for a bit more and then we'll take a break. So what happens when water molecules bounce to surfaces? So the water touching the edge of a glass or this box is going to be reduced in activity. And then if I have a protein, this blob in the center, the water touching that protein is also reduced in activity because now it's paying attention to the protein as opposed to water that's free in solution. So I can kind of simplify this by using this model where the lighter the color, the higher activity, and then the lower color or the darker gray corresponds to lower activity water, and shows us how when water is interacting with surfaces, it can decrease the activity. And then that's what happens when water is in bulk or when water is touching something hydrophilic that likes to make a hydrogen bond with it. But what happens if you have something like butter or something hydrophobic? When you have that, the water that's touching that can't form good bonds with the fat or the hydrophobic surface. So it's almost like it's increasing activity. It wants to diffuse out of there faster. It is frustrated. And this is kind of why on hydrophilic surfaces, the water will spread out and attach to it. But on hydrophobic, it wants to bond with itself rather than bond with the hydrophobic surface. So that kind of covers this rough idea of what happens when water is touching something, and it introduces this idea of kosmotropes and chaotropes. And kosmotropes are things like the hydrophilic surface, and they're defined as water structure enhancers, and chaotropes are kind of like the hydrophobic surface, where they're defined as water structure breakers. And to be sure, these words are models. There's no real mathematical definition. These are kind of hand-wavy arguments for kosmotropes and chaotropes. And so I'll talk a bit about them, but they are not rigorously defined yet. So understand kosmotropes and chaotropes. I like this paper, The Influence of Kosmotropes and Chaotrope Salts on Water Structural Relaxation. And at the baseline or in bulk water with no salt, water diffuses freely. So if you have one water molecule and wants to hang out or choose to hang out with one molecule or the other, it has no real preference and can diffuse freely. But then when you add salts like potassium chloride, which is a weak chaotrope, they find that water diffuses faster in the presence of potassium chloride. And this is really peculiar. So potassium is a charged salt, but and so the free electron pair on water is slightly negatively charged and would be attracted to the positively charged potassium, but it's not a dense enough charge to be more attractive than a nearby water molecule. So a water molecule would prefer to hang out with water than it would with potassium. And that's why it's a chaotrope, is that it kind of breaks down the structure. Sodium is the opposite. As a kosmotrope, it enhances water structuring and causes it to diffuse more slowly than bulk water. So in this case, sodium is smaller. It's still a +1 charge, but because it's a smaller electron shell, then it's a more dense charge, and water would rather its lone pair interact with sodium than with a nearby water molecule. And this is what caused it to be a kosmotrope. So this is relevant to us in biology because intracellular salt concentrations, like in a neuron, in the axon, is about 150 millimolar potassium, and inside the cell is only 10 millimolar sodium, but outside the cell is 150 millimolar sodium. So the cell likes to keep potassium inside and sodium on the outside. What does this mean? That when the electrical signal, called the action potential, travels down the axon, it's exchanging potassium on the inside for sodium on the outside. And that's what's actually being traveled down. So the proposed mechanism to describe this is these voltage gated sodium potassium pumps, where when the voltage changes between the inside and out, it recognizes that, which leads to a conformational change, which will open a channel and let the sodium flow in. But with this idea of chaotropes and cosmotropes, it offers a little bit of a different explanation for what we're seeing here, or different interpretation. And this is something that's been proposed by others. The scientists in Japan wrote this paper, Saltatory Conduction as an Electrostatic Compressional Wave in the Axoplasm. And what they've found is that if you have unmyelinated nerves, then there's these, the voltage gated ion channels work as expected, and they can sense the voltage nearby and continue the action potential. But when you have myelinated nerves, the voltage gated channels only exist in between the myelination, and the myelination can be hundreds of microns apart. And this author argues that that's far too far to recognize a voltage difference. So there must be some other mechanism that communicates the action potential more than just the voltage difference, according to their model. So one way to look at this is that when you have the sodium wave traveling down, it's actually changing the viscosity. So as it's changing down, it can interact differently with the protein, and you have a difference of surface tension almost when you have sodium versus potassium on the inside. And for this to be relevant to this, I need to prove to you that this can actually influence protein motions, where when sodium is on the inside, it'll move differently than when potassium is on the inside. And to do that, I'm going to borrow from this idea of Hofmeister effects on activity and stability of alkaline phosphatase. So the Hofmeister series is was a theory proposed by Hofmeister in the 1800s to describe how some salts would actually increase the stability of proteins and other salts would decrease the stability of proteins. And the salts that increase the stability also overlap with cosmotropes, and these salts that destabilize are chaotropes. So when they were looking at the initial activity of the enzyme, how fast it worked, compared to the salts, they found that the chaotropes cause the enzyme to act faster. And this makes sense because the breaking up the water bond structure would allow the protein motions to occur more easily. Whereas the cosmotropes decrease the enzyme activity, which would be in line with inducing structure and kind of rigidifying the protein. And so this is a simulation of water on a protein surface. And I'm trying to share that not only does salt share how water moves, but the protein surface itself can influence how water moves. So the closer the water is to the surface of the protein, it's colored blue. And the blue water molecules, they diffuse more slowly than the waters farther away in red and orange. So if they're just highlighting the blue ones, you can see that the interfacial water, the ones closest to the protein, they move much slower. And then the farther out you get from the protein surface, the more it returns to the bulk water behavior. And my favorite demonstration of protein's ability to structure water or play with water is in creatures that live in freezing conditions like this snow flea. So this snow flea will make antifreeze proteins, and these antifreeze proteins will actually bind to growing ice crystals. So as an ice crystal might grow in their cell, if they let it run away, it could rupture their cell membrane, like if you let spinach freeze in the freezer, and this is no good. So these antifreeze proteins will bind and stop the growth of that ice crystal in the cell. And in this paper, high water density at non-ice binding surfaces contributes to the hyperactivity of antifreeze proteins. These authors show that antifreeze proteins have an ice binding surface, and this ice binding surface seems to structure the water to make it more low density, and that looks like ice, so that orients the ice binding surface to the ice crystal, whereas the disordered water behind it kind of provides this pressure to push it there. So now what I've shared is that there's water behavior alone, and the anomalous properties by itself, and how this is explained by the two-state water model, and indicates that liquid water has some kind of structure maybe. And then whenever water is touching something, it actually changes its behavior, and things are either cosmotropes or chaotropes for structuring or destructuring the water around them. Now the next thing I want to talk about is what happens when water stops touching something. And I'm calling this to precipitation, when it drops out of solution. And I will talk about the law of matching water affinity. So when you have a folded protein or an unfolded protein, it's in a sense continuous with the water around it, but then if it folds, it pushes the water away from itself, and it kind of is like precipitated from solution. So a folded protein would be maybe even considered precipitated from solution compared to an intrinsically disordered protein. So when we're talking about proteins and denaturing them, causing them to fall apart, this person Collins, Dr. Collins poses looks at the Hofmeister series for stabilizing and destabilizing, or looking at cosmotropes and chaotropes. And Dr. Collins wants to propose in his 2019 paper, the behavior of ions and water is controlled by their water affinity. And what he means by this, from the data they shared, this is an enzyme half-life that they purified from E. coli. And when they keep the enzyme in a cosmotrope solution, it's much more stable, again stabilizing, so it's active for longer, but in a chaotrope, it falls apart and denatures. So what Collins wants to understand is how these are interacting with each other to create this effect. And he's going to argue that it's based on water affinity. So to do this, he's going to look at how different ions and different salts run through a Cephredex column. So this is just a size exclusion experiment where they have Cephredex beads, and these beads have pores, so small molecules can get lost inside these pores, and it'll take a long time for them to diffuse through the column, but larger molecules that can't get lost in these pores, they go through much quicker. So as the column progresses, the large molecules go through faster and the small ones go through much slower. And what Collins finds is that cosmotropes, they have a higher affinity for water, they want water's attention, and this gives them a larger radii, so they move faster through the column. And then chaotropes, because they have a lower affinity for water, they're actually kind of smaller, and they have a smaller radius that moves more slowly through the column. So what he's doing is running a column with 0.1 molar sodium chloride, and then he's going to run these different molecules through. So dextran is very, very large, so it goes very, very fast through the column. And then these chemicals take longer. So THO is actually tritiated water, so that's his control for straight water. And they find that fluorine moves quite quickly through the column. So between dextran and water, there's fluorine, and the water, fluorine is really, really electronegative, really, really densely charged, so the water is paying a lot of attention to it, and it forms a couple of hydration shells around it. So oddly, one of the smallest anions actually has the largest hydrodynamic radius and flows the quickest through the column. Chlorine only has a one shell, and so it appears as a smaller ion going through the column. And then iodine is a much larger atom, but it has a it's much more weakly interacting with water, so it doesn't really have a strong hydration shell. And this actually causes it to get pushed to the side, because iodine would rather not be in water, and that's why it takes longer than it does for water to go through. That's why iodine is so far behind, because Collins is arguing that's actually getting pushed out of the solution. And this is their measure of water affinity. So that's for anions. The same experiment or a similar experiment was done with NMR for positive cations. and they find that lithium has the largest hydrodynamic radius by NMR, and that cesium is the smallest. And this is, of course, kind of opposite of the size of the atoms themselves, where lithium is really small. And the interpretation of this is that lithium is so charge dense that it coordinates two shells of water around it, whereas sodium is only one, and potassium doesn't really even have, and cesium do not have a strong hydration shell. So their data is NMR, and then they model that as hydrodynamic radius for diffusion, and this matches what we saw with diffusion from neutron scattering earlier. So how does water affinity pair with the JUMP model? I argue that they agree because of this paper by the JUMP water model group called Water Reorientation Dynamics in the First Hydration Shells of Fluorine and Iodide. So the JUMP model again is that for this water to diffuse to the side, it's going to need to form a new bond and then break an old one, and this involves large rearrangements, angular jumps. Now what they find is that when they're looking at how the water density, so the hydration shells around water and these ions, when they're looking at just straight water or neat water, that there's this first shell where as you go out you run into a water, and then there's a hump for a second shell of water density, which is the second water molecule here, and then as you get farther out it just becomes density of one. It kind of becomes noise. Fluoride is different in that it has a really strong first shell and then a really strong second shell as well. But when they look at iodide, there's a strong first shell, but there's no real peak for the second shell. So iodide doesn't coordinate as many waters as fluorine does around itself. And then when they look at the orientation or time correlation of bound water, this for neat water, the more time that goes on, the less correlated the nearby waters are with each other. In fluorine, as time goes on, these waters are very coordinated with each other, indicating a very strong hydration shell. And in the case of iodine, it's actually lower. As time goes on, these waters fall apart and are less coordinated than neat water. So their data here is simulation, and the model that they take is that water jumps are encouraged around chaotropes. So something like iodine or a chaotrope encourages the reorientational jumps that they're talking about. And the overlap that I pose is that water affinity is similar to water activity. So the water around fluorine is really immobilized and very low active, whereas the water around iodine is higher activity. And this agrees with neutron scattering, their column experiments, and the computation to pose the JUMP model all agree with each other from these different groups to help build this idea. And then what we're really wondering is how salts precipitate, right? How do things fall out of water? So for things to fall out of solution, for salts to precipitate, you have to have these two hydrated salts come together, and when they do, you lose water, right? It pushes the water out of the way to allow the salt ions to interact with each other. And this can form a direct ion contact pair, or it can also form a solvent separate ion pair, where there's always a water between these two charged ions. So how does this make sense? Well, when you have a strong cosmotrope that's negatively charged or a positively charged cosmotrope and a negatively charged cosmotrope, they prefer to form a direct contact pair. So they both precipitate out and release the waters and touch a chaotrope, and a chaotrope is similar, a positive chaotrope and a negative chaotrope. They both push the water shells out, but then when you get to mixing a positive chaotrope with a negative, or a positive cosmotrope with a negative chaotrope, they form a solvent separated ion pair. And the way to interpret this that I take is that the charge on the negative ion, the negative chaotrope, isn't strong enough to displace the water that's strongly attracted to the cosmotrope. So it's much harder to get these to precipitate out when you have cosmotrope and a chaotrope in solution, they tend to stay in solution better, right? So if you have positive cosmotrope and negative cosmotrope, they're less soluble, and then again, chaotropes together are less soluble. But when you have salts where they're across a cosmotrope with a chaotrope, it's much, much more soluble. And this is actually when we're learning precipitation in general chemistry, how we're deciding what's being precipitated. So when you add lead nitrate, where lead is a chaotrope and nitrate is a cosmotrope, with sodium iodide, where sodium is a cosmotrope and iodine is a chaotrope, then the two chaotropes will precipitate out as a direct contact pair. So Collins' law of water affinity actually helps us understand how salts precipitate out of solution, and the question is, how is this relevant to the cell? So what's really curious about the cell is that macromolecules like RNA and DNA, these polymers are really, really strong cosmotropes. The phosphate backbone is very, very attractive to water and forms it and is a very strong cosmotrope. So the DNA has a very strong hydration shell around it. Proteins the negative charges on proteins are carbonyl groups. So aspartic acid and glutamic acid are both cosmotropes. Water is very strongly attracted to both of these, which means that all the large polymers in the cell are negatively charged cosmotropes. And this means that when we look at colloidal theory, that if we're looking in the cell, these negatively charged large particles tend to push away from each other. And this is really helpful for the cell because we don't want precipitation to occur. We want all of the proteins and the RNAs to be freely flowing over each other unless otherwise. And then you can add countercharges to get them to aggregate if you want. So this idea is experimented and played with by this group that has this paper, The Hydrogel Nature of Mammalian Cytoplasm Contributes to Osmosensing and Extracellular pH Sensing. And what they did is they permeabilized the cell membrane, and this kind of allowed some water in. But then they would change the pH or change the osmotic pressure and then see how the volume of the cell changed over time. And what they found was that whenever they deviated the pH from 7.4, the cell's volume decreased. And one way to interpret this is that at pH 7.4, which is our blood pH, nice and neutral, all these things are negatively charged. The strong cosmotropes are pushing each other away. And then as you get some positive charges from the lower pH, or you start to affect this ability to repel each other, and then they'll start to aggregate. Then we're wondering, what about the positive charges on these macromolecules? So proteins, the positive charges on proteins are arginine and lysine. These are both chaotropes because the one positive charge, is kind of distributed over a large number of atoms on the side chain. So this means that in the cell, all of the negative charges are strong cosmotropes, and then all of the positive charges are chaotropes. And this is firmly in this more soluble area, right? So this is one way by which this chemistry is really relevant to understanding how molecules are organized in the cell, because it kind of maintains the solubility, right? It seems in retrospect it kind of has to be this way. And then what about the salts in the cell? So intracellular normal salt is potassium chloride, or potassium rather, which is a positively charged chaotrope at about 150 millimolar. And again, it satisfies this where it's a positive chaotrope, so it allows all the molecules to move nicely with each other. But then when you think of a action potential where you're replacing that with sodium, or triggering muscle contraction where you're releasing waves of calcium into the muscle, now you're adding a very, very strong positively charged cosmotrope, right? Less soluble. So we can almost think of the action potential or the contraction of muscle as this weird precipitation reaction where you now add this really, really insoluble ion pairing. where the calcium or the sodium could maybe interact with the cosmotropes on the polymers, giving us a different view or a different lens for biology. And this kind of means when we're looking at things that interact with DNA or RNA, if you have, it's always going to be a cosmotrope and a chaotrope, so that they should always maintain a water between them and move nicely over each other. So now I've covered how water behaves alone in a glass, what happens to water when it's touching something, and how things can be either cosmotropes or chaotropes, and then what are the conditions when things precipitate, and how the law of matching water affinity can help us determine what precipitates. And so with this, I have this idea that everything in biochemistry is actually a condensation reaction.
Question is, do you know the density of, way back when we were talking about the liquid-liquid phase separations with the protein, you said, you have like the more structured water, and then in the phase separation ones, I was curious like what the density was, comparison between the phase separation and the LLPS sorted protein in it versus not.
So I think that there's a few answers. First of all, like usually the protein and macromolecule concentration's higher. So then the question is like, is the remaining water more or less dense than in the bulk? Is that what you're wondering?
Yeah, I guess I was just like curious about like, so like the water's less dense, but I was like, is the protein making it more dense or something?
Yeah, the protein's making it more dense. Yeah, but yeah, when I talked about the terahertz thing, indicating more tetrahedral water, that kind of is like perfectly coordinated water, and ostensibly the water itself is in like the less dense kind of like almost low density type because it's now better bonded. But the actual phase itself is more dense usually because there's more macromolecules, and those are usually more dense than the water.
Nice. That's interesting.
Yeah.
So it doesn't float.
Depends on what you put it on. I think that, yeah, I kind of am wondering if there's a way to get a granule in the cell that's like less dense than the surrounding cytoplasm, and I'm not sure. I haven't found a way to like cheat that yet, unless you're using like a liposome, but then you're cheating because it's lipids and not water. So
Precise.
Yeah, perfect. Yeah. Let's see. Any other questions or comments? Curiosities?
What's your favorite story of what you just went over?
I really like the action potential one. The part that I just covered, I really like where it the difference between potassium and sodium I would never pay attention to in my buffer making, but now it's just so clear that like it's so seemingly natural how the cell organizes itself, like what chemical and functional groups it's using and why. It's kind of beautiful with this lens to me now, right? Like it has all these places. These aren't just haphazard, and to me that's really striking.
Very true.
Yeah. What about you? Is there any favorite little tidbits that I've gone over?
I really enjoyed the sulfur, like the series where water like pops up and you have the H2S and like the other one. I thought that was something new I hadn't seen before, and I thought that was like just that little plot was nice comparison.
Yeah, I've been trying to search for all those like convincing little stories that show that water is different. It's like hard to prove quickly. Yeah.
So, I really like the part that you talk about how water behave when they touching something. I'm just curious. Like you use those dash line to show the movement of water touched to something. Are those dash line real, or it's like just a presentation?
Let's see. Maybe I want to make sure that I know what you're talking about. So stop me when I like get it. Go ahead.
You show the water behave like a water with different activity when they touch to something. I'm wondering, are those movement of the movement of water real, or you just put the dash line there said okay, this is like less activity, those are high activity.
I,
Like touching a glass? Or...
I let's see. I'm trying to remember the dashed lines that I had when it's touching glass. So
Yes, here.
here, okay.
Can you go? Yes, those dash line under the water molecular.
Yeah.
Are those dash line a real measurement or just?
So the dash lines, like if I was to look at the diffusion or like where these molecules are in 10 nanoseconds, I would say like this, because this is immobilized on the side of the glass or on a protein, it diffuses much less. So this is made up, but it is trying to capture something that is real. And then the first shell of like bound water to this immobilized charge, it moves less distance in the same amount of time. So this water will like come on and off, but in the same amount of time, it will move less than something that's farther away. So this is kind of made up, but it's very much like there's really good data to support that this is true.
Okay, yeah, good to know that. I mean, it will be super cool if you can see the movement of water.
Agreed. Yeah, the movement of water is all inferred, and usually this comes from simulations. And simulations are on the border of like data and model, because all of our water simulations are like good for some things but not great for others, which means that they're imperfect. So it's uh, it's probably how it's going down. It's probably reliable, but we can't really look at the water directly, so it's really hard to actually like measure that. And so you're right to like ask about that.
Thank you.
Sure.
Okay, really great talk so far. So this is just thinking about the talk we went to today.
Yeah.
Does heavy water, that containing deuterium, behave the same way as what you're describing here?
Yeah, it's a great question. It's super complicated, unfortunately, because people do deuterium exchanges like heavy water all the time to do water experiments, and a lot of times deuterium has a stronger hydrogen bond, and this means that like it will diffuse slower. So kind of going back to what I was sharing with Jinjin, like heavy water will diffuse slower in the same amount of time because its hydrogen bond is stronger, so it like won't let go as well. But if you drink heavy water, you will die, right? And this kind of means that these things aren't right, like they aren't chemical. Well, if you drink enough, if your body becomes heavy water, you don't get to live. And what this is alluding to and is coming out recently is that there's some subtle differences in like the quantum behavior with the bonds, basically, is where you have to look for this. And because of that extra neutron, it changes the behavior of the water in a way that's truly different, and it's not exactly the same as the normal water. So it's like reasonable to replace with, but it's not perfect. And what's cool about it is because the stronger hydrogen bond, you can see these trends like hold. So if you replace it with heavy water, you should expect to see things move slower, and people use this a lot, but it's not exactly the same.
Oh, thanks.
Okay. Any other questions, or just yeah. I like this next part. It's not as practiced, but it's this everything is biochemist. Everything in biochemistry is condensation. So this is kind of where I'm throwing my own spin on these things and reinterpreting some common things through this lens. So we left off with salt and ions, right? When they come together and precipitate, you're releasing these waters into solution. So going back and forth is a condensation reaction. Well, when you have ATP and magnesium in the cell, so this is the same thing, that magnesium and ATP, they're both strong cosmotropes. So in a sense, they precipitate each other out of solution. And when you have the ATP magnesium complex, that itself is almost a precipitate. Right? And when you have the hydrophobic effect, two hydrophobic things coming together, that also releases water, which is a condensation reaction in a sense. Same thing for protein complexes coming together. And protein folding even. So in protein folding or protein structure, when you're making the primary sequence, so you're stringing these amino acids together, the very creation of that string releases a water molecule, to go backwards and forwards. And then when your protein string is now folding up to create secondary structure, as you form beta sheets and alpha helices where it hydrogen bonds with itself, those hydrogen bonds are, as it's recovered with the protein, releases the hydrogen bonds with the water, right? So you're letting water go as you're folding the protein. And then as a secondary structure comes together in tertiary structure, that continues the same as well, where you're now releasing the water molecules that are on the faces. And when you come together to form a quaternary complex, same thing as well. The interfaces between them are releasing, you're pushing the water molecules out. So I really like this one study that's looking at this kind of intrinsically disordered to alpha helical transition. And it's it's titled Osmotically Induced Helix Coil Transition in Polyglutamic Acid. So their IDR is just polyglutamic acid. And when they measure it or observe it with circular dichorism, they can see helix forming as they increase the amount of PEG, which they say is an increase in osmotic pressure. And so at neutral pH, they don't form a helix, but then at slightly acidic pHs, as you increase the osmotic pressure, you then induce this normally disordered region into actually forming a helix. And they say that our results show that osmotic pressure significantly affects the protein folding equilibrium. We therefore suggest that to study protein folding in vitro, the osmotic pressure, in addition to pH and salt concentration, should be controlled to better approximate the crowded environment in cells, which I think is really important for when we're doing biochemical experiments in the test tube to keep in mind.
Michael,
Go ahead.
can you go back one slide?
Yeah.
Now, you said when you lower the pH, is this from pH 5 to 7 down to 5? Is that what you're saying?
Yeah. So 7 is
Okay.
here. Yep. Yeah.
Good.
That's right. Yeah. And so all these protein motions and all these common things that happen in biochemistry that we think of are all just playing this weird accounting game with water, is what this is kind of telling me. So when I was trying to endeavor to understand how stress granules might concentrate proteins and then lead to amyloid fiber formation, at every step we're going from monomeric FUS to liquid or to phase separated FUS, you lose some water for these stickers to interact, and then when you create a solid amyloid fiber, you've now gotten rid of all of the hydration shell of that protein. So when I want to try and understand stress granules and how it forms amyloid fibers, this is the question I'm really asking, are what are the conditions where I can create a favorable environment to get rid of a hydration shell of a protein, right? And I can't answer the amyloid question today, even though I started with it, but it's still cool to see where this goes with biology, I think. But to get to that, I kind of need to answer this question, what is water activity and what is osmotic pressure? And the reason I ask that, or feel the need to start to define that now, is that they're kind of these loose definitions. So if you have this normal U-tube with a semi-permeable membrane, and you put salt on one side, that decreases the water activity, so then the water on the free side, or the pure water side, wants to flow over, and for the same reason why the water activity is decreased is the salt kind of immobilizes some water. And this is called osmosis, and I'm borrowing Wikipedia definitions to read, not to be really boring, but just to be really, really explicit in what the established ideas are and what I'm saying is going on, and how they agree. So osmosis is the spontaneous net movement or diffusion of solvent molecules through a selectively permeable membrane from a region of high water potential to a region of lower water potential, in the direction that tends to equalize the solute concentrations on two sides. That's from Wikipedia to describe osmosis, which is what's happening here, where the water will then flow to the salty side to try and equilibrate concentration, and that osmotic pressure becomes this difference, right? At a certain point, the water can't fight gravity to go equalize this side anymore, and so the osmotic pressure is the pressure that you need to push water back, right? And the Wikipedia definition is osmosis can be made to do work. Osmotic pressure is defined as the external pressure required to be applied so that there's no net movement of solvent across the membrane. Osmotic pressure is a colligative property, meaning that the osmotic pressure depends on the molar concentration of the solute, but not on its identity. And I'm going to, first of all, highlight that osmosis can be made to do work is really important, but that also this definition from Wikipedia, we're going to have to figure this out, right? I'm butting heads with it on some way. And what that means is that this, so the osmotic pressure is an ideal parameter, or is basically the osmotic pressure plus A plus something, and this is the formal definition for osmotic pressure, where A is an empirical parameter. The value of the parameter A and the parameters from higher order approximations can be used to calculate sizer parameters. Empirical parameters are used to quantify the behavior of solutions into ionic and non-ionic solutes, which are not ideal solutions in a thermodynamic sense. And the only thing I want to translate this to really is that these are empirical parameters, which means that we measured them. And so we're kind of just measuring, like osmotic pressure is this fudge factor, right? It is this like dark matter of biochemistry, in that we know that the gravity is off, and that's why we need dark matter to hold universes together. In the same way, we need osmotic pressure to hold these concepts together, but this isn't giving us a molecular level description of what osmotic pressure or water activity is. It's just describing general behaviors, right? So on the mechanism of osmotic pressure from Wikipedia, it has the mechanism responsible for driving osmosis has commonly been represented in biology and chemistry texts as either the dilution of water by solute, resulting in lower concentration of water on the higher solute concentration side of the membrane, therefore a diffusion of water along the concentration gradient, right? Or by solute's attraction to water, resulting in less free water on the higher solute concentration side of the membrane, and therefore net movement of water towards a solute. So just describing this situation, both of these notions have been conclusively refuted. So this generally describes it, but we know it's wrong. Why is it wrong? The diffusion model of osmosis is rendered untenable by the fact that osmosis can drive water across a membrane towards a higher concentration of water. Right? So you can choose a salt where it's going to go the opposite way, where it's, the water will be pushed towards a higher concentration of water, which is kind of backwards. And the bound water model is refuted by the fact that osmosis is independent of the size of the solute molecules, a colligative property, or how hydrophilic they are. It is difficult to describe osmosis without a mechanical or thermodynamic explanation, but essentially there is an interaction between the solute and the water that counteracts the pressure that otherwise free solute molecules would exert. One fact of note is that the heat from the surrounding is able to be converted into mechanical energy or water rising. So the main takeaways for me is we're lacking a very explicit mechanical explanation of what osmotic pressure actually is. So when it's talking about these particles being like colligative properties, meaning that it's just a number of particles, we kind of know that's wrong. And this is shown by aqueous alkali halide solutions. Can osmotic coefficients be explained on the basis of ionic sizes alone? And what they're saying is that they look at these different cations, these singly charged positive ions, compared to, with bromine, which is kind of a chaotrope. And they see that lithium, which has the largest hydration shell, is exerts the most osmotic pressure, has the highest osmotic coefficient at these concentrations, whereas cesium has the lowest. So this kind of follows what we were talking about earlier, where the more structured water that you have around the ion, then the higher osmotic pressure or coefficient that it exerts. So it's kind of adding to the salt, and some structuring, some playing with the water does matter. It's not just the number of particles that you have in solution, which, you know, is at odds with these definitions sometime. So what I'm going to do then is pose my own definition that it's fine to refute, that water activity is the likelihood of water switching H bonds to another molecule. So kind of this same figure here, how likely are you to hydrogen bond? If you're a chaotrope, you're likely to hydrogen bond with another water molecule. If you're a cosmotrope, you're unlikely to bond with another water molecule. So lower activity, higher activity. And I think this is supported by a jump model, the law of matching water affinity, and the two-state water model, and that osmotic pressure I would describe as some degree of structuring of water, which was explicitly said was not happening. So these are kind of these terms I'm going to be playing with moving forward, that I've defined what water activity is and osmotic pressure is, and it is fine to disagree with that. And that even proteins will play with this too, this degree of structuring of water, like the antifreeze protein. So, yes, agreed. So now, now that I've kind of given a definition of water activity and osmotic pressure, now let's go on to understanding how liquid-liquid phase separation and condensates might be functions of water or condensation reactions. So condensates and phase separation biology as condensation, and what I'm getting at is co-aspirates and hydration. If I'm looking at stress granule formation in the cells, like by adding sodium arsenite, we might think of this in the sense of like a stress pathway, like a chemical stress pathway. And this is useful for understanding some things, but the stress granule forms faster than you can conduct these signals usually. So you can form stress granules in minutes, where usually to get changes in DNA expression, this could take like 24 hours or a long time. And furthermore, I can create stress granules without arsenite or just by dehydrating the cells with osmotic pressure. So if I add 250 millimolar sucrose, it causes these cells to form stress granules, and it causes them to shrink, to decrease their volume. So as opposed to thinking of stress as this like activated pathway, if we're looking at this mixture of like intrinsically disordered proteins and globular proteins in the cytoplasm, then liquid-liquid phase separation would be these IDRs coming together. And this can be done through many different ways. Again, adding this just shrinks the cell volume, and that can create the same effect as the sodium arsenite poison. So to understand this, I could see that just changing the volume of the cell is enough to get stress granules or phase separation to occur. And I can also change maybe the water activity. As you decrease free water in the cell, it becomes lower activity water. And then when you get phase separation, you get these proteins, these intrinsically disordered proteins and RNAs together in a stress granule. I'm saying that there's an implicit assumption that these stickers are now sticking with each other. So for the sticker and spacers model, I'm saying that the stickers are now interacting with each other over water in this environment. And that means that this is kind of a contact ion pair, right? This is pushing water out of the way for the stickers to be able to come together. So that would be like arginine associating with RNA or something like that. And then when we're wondering what's happening to the water in this situation, we can refer back to this work that I cited earlier, the key rule of solvent and condensation, mapping water and liquid-liquid phase separated FUS, where they find that the water was more tetrahedral in the phase separated condensates, meaning that it's more coordinated, right? More ordered, with more ideal bonding. So the phase separated regime here, I argue, has a different hydration than before in the bulk. And I think that this is supported by terahertz, the infrared models, and also this, again, this paper, osmotically induced helix coil transitions in polyglutamic acid, where just increasing the osmotic pressure by PEG can fold this protein at low pHs. And this aligns really nicely with this 2017 science paper by Alberti's group, where they find that the phase separation of a yeast prion protein promotes cellular fitness. So they look at this prion protein SUP35, and when they starve and energy deplete these cells, they actually decrease their pH to like 6, and they find that they get stress granules to form. And they say that in SUP35, it's the M domain, which is got many negative charges, like a polyglutamic acid, that is important for sensing this. And they say, thus, pH sensing of SUP35 is facilitated by its charged M domain through protonation of glutamates. We propose that the high density of acidic residues in the M domain causes an upshift of the pKa value of glutamic acid, where it now becomes, instead of like 3.5, 6.5, or something sensitive to yeast. But this can also just be seen as increasing the osmotic pressure inside of the granule, leading to the same effect. This is also seen for TDP43. So the Fauci group looked at TDP43 alpha helical, and they have this paper, TDP43 alpha helical structure tunes liquid-liquid phase separation and function. And what they find is that the low complexity domain of TDP43, which is a protein that forms amyloid fibers in ALS in some cases, as this reversible alpha helix in it, and they find that in the phase separated droplet, the alpha helix is formed. And I think, again, this is another mirror of this idea that in the higher osmotic pressure or the lower water activity environment of the granule, you favor this alpha helix that's formed. And I think that all of these together kind of overlap with each other in indicating that there is a unique hydration environment inside these coacervates or inside these phase separated droplets, and that it will be different from the bulk or the outside, right? And that there is an entropic penalty for organizing the coacervate. So because these things can't diffuse around freely anywhere in the cell anymore, that's an entropic penalty. And we find that the protein and water motions are slowed inside of the condensate. This, I argue, is paid for with better bonds. And so as you get to be close together and you're overlapping your hydration shells, you now get to have better chemical bonds. And these slowed motions plus better bonds are kind of this hallmark of low density water, which aligns with this idea of more structured or tetrahedral low density water manifests as higher osmotic pressure or lower activity in some level. And that this higher osmotic pressure fights the desired flow. So water wants to flow from high activity into low activity, and then it's exerting, this granule is exerting some kind of pressure that fights that desired flow of water inside of it. And so this I cite as some evidence or some ideas how the cell can maintain unique hydration environments in phase separated or coacervates. And so the question is, does this exist in biology anywhere? Can I find examples where there's compartments of cells with unique water activity and osmotic pressure? Alright, so cool. To do this, I'm going to invoke this YouTube experiment again with a semi-permeable membrane where the water flows to the saltier side until the osmotic pressure equilibrates. But if you get rid of this U2 and you don't allow the water to move, then you have to rebalance in some other way. So I'm going to argue that this is kind of the same setup as where to fight the waters wanting to go to lower activity, osmotic pressure fights back. And I argue that this is the same setup as the nucleus versus the cytoplasm in the cell. So the nucleus is a semi-permeable membrane that lets water and some salts and other molecules through, but big proteins and molecules, they can't go through it. So therefore, it kind of replicates the same behavior, I oppose, and that this means that the cell nucleus is a unique hydration environment. And now I'm going to try and show that that might be the case. And I'm not the only one posing this. This is an idea that's taken from this theory paper called Balance of Osmotic Pressure Determines the Nuclear Cytoplasmic Volume Ratio of the Cell, that was published in 2022, which is saying the same kind of idea. And it borrows from a study done by this group where the relative densities of cytoplasm and nuclear compartments are robust against strong perturbation. And this means that as the cell grows, it maintains the relative densities of macromolecules inside and outside of the nucleus, in the cytoplasm and in the nucleus. And if you do certain drug treatments, you can change the amount of free molecules by depolymerizing or not the cytoskeleton, and it still maintains a relative different densities, which kind of mirrors this semi-permeable membrane behavior. So here the data is that it maintains these relative densities, and that the nuclear cytoplasmic, and then the model is that the nuclear cytoplasmic ratio reflects a difference in osmotic pressure between those two compartments. And this is further compounded by studies where these groups have looked at small dyes that have a single hydrogen bond acceptor. So this dye has a single nitrile group, which is really, really chemically unique, with a single lone pair that can accept a hydrogen bond. And they see that for that chemical group, when there's less water, it redshifts, and when there's more free water, it blue shifts that chemical group. So if they can just measure this dye's spectrum, then they can see how that influences or ties back to the hydration environment. And what they show is that there is indeed a difference. They can see the nucleus with a different chemical shift of that bond than the dye in the cytoplasm. And they state that a marked and consistent blue shift is observed in the nucleus relative to the cytoplasm. Among 98% cells showing an increased solvation and hydrogen bonding in the nucleus. So their dye shows an increased solvation and hydrogen bonding in the nucleus, which kind of indicates higher activity water, more available water in the nucleus. And this result was supported, or identical result, from another paper titled Optical Mapping of Biological Water in Single Live Cells by Stimulated Raman Excited Fluorescence Microscopy. Does almost an identical type of experiment, but they add one more of this SRS signal, where they find that water density inside live HeLa cells measured by SRS microscopy, and the results show that the nucleus is the red line, the cytoplasm is the blue line, that the nucleus is more dense with water and the cytoplasm has less. So again, supporting that from the data being the fluorescent shift from a probe, that they find different hydrogen bond strengths and densities in the nucleus versus the cytoplasm, which I argue supports this theory that the nucleus is its own unique hydration environment in the cell. And that then I would extrapolate that stress granules would be the same version of this, where they're almost in a semi-permeable membrane. Some molecules like to be in the stress granule, other molecules like to be outside. And that nucleus and condensates have stable, unique hydration environments, as dictated by unique environments for osmotic pressure and water activity. And yeah.
Sorry, that was weird what I was doing. Could you go back to the difference figure? Yeah.
Yeah.
I mean, not that I know how to interpret that effect size, but off the cuff, looks like a really small one.
So off the cuff, you're totally right. It's super small because the difference is ostensibly, really small of how much distance you get from hydrogen bonds, like an optimal versus a suboptimal. It's really, really marginally small, but it seems like they reliably measured that difference, and I think that's what matters to me. But it's super minute, but I think it is above error. Yeah.
For sure, I think it's definitely significant. I guess I'm kind of trying to understand more intuitively what a 0.02% difference in signal means.
Yeah. I don't actually have a good answer for you on this one, because this type of microscopy, I'm really parroting what they're saying here, but I don't have a firm understanding of the theory that they're working from. I'm hoping to pick that up. Other stuff you could ask these questions I have better answers for, but this one, like, you're right to kneel that one. I don't have a good answer for it. Yeah. I wish I knew more.
Thanks.
Yeah.
Me too, buddy.
Right? That's what we're missing. Cool. And so this idea of the nucleus and the cytoplasm having their unique hydration environments, but that they talk to each other, I think means that this nuclear cytoplasmic volume ratio balance might be relevant to biology. And these titles are from a 2006 and 2007 paper from the same person, Dr. McIntyre, where they pose that cell hydration as a primary factor in carcinogenesis, a unifying concept, and increased cell hydration promotes both tumor growth and metastasis, a biochemical mechanism consistent with genetic signatures. So these are models, these are review articles, but what this person is proposing is that when you're a stem cell, the nuclear cytoplasmic ratio is like 1. It's the nucleus is similar in size to the cytoplasm, but then when you're very differentiated, like a neuron or a muscle cell, you think that you now get super huge, long, like tons of surface area. Your cytoplasm becomes really large relative to the nucleus, nuclear size. So differentiation would almost be going down, right, the nucleus cytoplasmic ratio, and then cancer would almost be going up to be during oncogenesis. So the stem cell, and the differentiated cell having different ratios. And in fact, people have gone a little bit farther to show this. So in this paper titled Role of Intracellular Water in the Normal to Cancer Transition in Human Cells, Insights from Quasi-Electron Neutron Scattering, what this group does is they look at scattering of cytoplasmic water in healthy cells and cancer cells, and they find that the dynamical profiles obtained for cancer versus non-malignant cells showed a clear distinction between them. Tumor cells displaying significantly higher plasticity relative to the non-cancer ones and enhanced mobility of intracellular water, thus being a potential hallmark of malignancy. So what they're saying is that, as the cancer cells look a little bit more kind of like those stem cells where there's more water, and the water is more freely moving in the cytoplasm, supporting this idea. And in fact, people are now trying to use water environments to diagnose cancer or tumor. So terahertz refractive index-based morphological dilation for breast cancer carcinoma detection, which is using the terahertz hydration to identify hydration environments for associated with cancer. And then another using attenuated total reflection Fourier transform infrared spectroscopy to distinguish between melanoma cells with a different metastatic potential. So now engineers are trying to use these roundabout ways of looking at light for hydration to actually identify cancers and tumors, which I think, again, supports this idea that unique hydration environments and for cells is really, really important. And so I'm posing that free water is different between cells. So like if you're a stem cell or a differentiated cell or a cancer cell, then your amount of free water and the mobile water will behave differently. And that this extends to compartments. So in stress granules, there should be less free water. And that this different water activity and osmotic pressure between cell compartments, and this would cause different pressures to precipitate. So again, increased pressure in a stress granule where there's less free water and more structured water. Now, I think that I want to skip ahead some part because this is long, and I'm just trying to show that I have this Actan dye, which can actually identify or granules in the cell by having these like blue shifted compartments. And I think that it's been 2 hours, so I think I'm going to wrap up just to save everyone's time. I'm very appreciative of everyone being here. This last part is kind of cool. I'll run down my favorite highlights, and if you have questions, I can explain more. But basically, this Actan dye I'm now using at St. Jude to try and identify unique hydration environments. It should be fluorophore that's sensitive to that. And this group Bagatoli, in this paper, The Innards of the Cell, studies a water dipolar relaxation using the ACTAN fluorescent probe. They look at ACTAN signal from yeast, and what they do is they find when they synchronize yeast, so they starve yeast and then they add some sugar, and so their yeast is synchronized, and then it starts waves of metabolism or metabolic waves, and they see waves of metabolites. And what they find is that they get these waves of NADH over like 40-second intervals, and that they get waves of ATP concentration that also change over the same but opposite phase. So when NADH is high, ATP in the cytoplasm is low, and the ACTAN signal, which kind of corresponds to water behavior in the cell, tracks with ATP as well. So in synchronized cells that are talking to each other, they see that there's all these fluctuations of the water activity, the amount of metabolites, potassium levels, and ATP levels, and water polarization. And they use this to establish this theory of glycolytic oscillations and intracellular potassium concentrations are strongly coupled in yeast, Saccharomyces cerevisiae. And this allows them to build kind of a biophysical model of the cell, when it's almost like these waves are like the cell breathing, right? And so what does this kind of mean? They got the idea for this work based on this professor called Dr. Gilbert Ling, who proposed this thing called the association induction hypothesis. And I copied his Wikipedia profile, and that he actually made the electrode that we use for measuring action potentials in like the 1940s in neurology, and the association induction hypothesis, which is an alternative and controversial hypothesis to the membrane and membrane pump theories. The association induction hypothesis is claimed related to the properties and activities of microscopic assemblies of molecules. Sorry, that's not super useful, but it invokes the need of this structured water, and structured water is considered a pseudoscience on Wikipedia on this article. So again, at odds with my osmotic pressure. But what Dr. Ling noticed in the 50s and 60s was that when he would take frog muscle and cut open one end and then put it in a salt solution, that sodium wouldn't enter the muscle for much longer than expected by diffusion. So it's almost like the environment of the cell, of the muscle cell, excluded sodium and selected for potassium. And this was really odd because it kind of defied what we expect from diffusion, where we'd expect sodium to diffuse in freely, but it didn't. It took much longer than expected. And this kind of gave him the idea of a structured hydration environment inside cells. And this is a quote from the one of the inventors of the MRI machine. And it says, "On the morning of July 3 at 4:45 a.m., we achieved with great jubilation the world's first MRI image of the live human body. The achievement originated in the modern concept of saltwater biophysics, on which you are the grand pioneer with your classic treatise, the association induction hypothesis." So him thanking Gilbert Ling for his work so that they could come up with the idea for an MRI machine to look at these unique hydration environments. But then if you Google, is the association induction hypothesis true? The first hit is this Quora thing on Google. Why don't biologists ascribe to the association induction hypothesis? And you get a bunch of people saying that his work is not very reviewed, or it's just reviewed by a small series of journals. And they cite quotes by Gilbert Ling, where Gilbert Ling would say things like, "To keep membrane theory afloat, submicroscopic pumps were installed across the cell membrane. 45 years ago, this version of the membrane theory was also experimentally disproved. In spite of all these overwhelming evidence against the membrane pump theory, it's still being taught as verified truth in high school and biology textbooks today." Which is kind of a weird sentence when this Nobel Prize winner, Jens Saku, won the Nobel Prize for the first discovery of an ion transporting enzyme, the sodium potassium ATPase pump. So again, here's a structure of the ion pump that Gilbert Ling is kind of refuting. So this is why people typically don't like to or ascribe to his ideas, is that it seems clear that membranes exist and that they have pumps. But I think what's most interesting about his book on the association induction hypothesis is that he imagines a cell theory without the need to invoke a membrane. And I think that there's incredible intuition to be gained from that, and that's what this yeast group has been basing their theories on for looking at these. So again, I like this quote, all models are wrong, but some are useful. So with this in mind, I like to keep the things I like from Gilbert Ling, and one of them being his idea of the resting state of a cell being very ordered and so low entropy, where you have the potassium nicely bound to the cytoskeleton, and then water being ordered around that. And then when you do an active state like an action potential or a muscle contraction, what you switch to is a high entropy state where now you release these sodium or calcium ions, they switch with the potassium, and this breaks up the polarized and organized water layers, leading to this depolarized water. And that to recover from this, then you spend energy like ATP and the pumps to recover the low entropy resting state. So this is like resting, muscle contraction, and then you spend energy to get back to this. And this idea is very, very small, but talked about in some other papers, where they again pose this resting, highly organized state, and then this work cycle where you decrease order, and then you spend energy to go back to this highly ordered state. And this is what that yeast group was working off of to find these oscillations in yeast, where they find the dynamics of intracellular water constraints glycolytic oscillations in Saccharomyces cerevisiae. And what this paper is saying is that when they induce mutations in the yeast, if ever they leave this range of actin water polarization, the yeast can no longer do glycolysis. So the ability for yeast to do glycolysis means that it has to like operate within this narrow-ish range of water polarization and water of activity and osmotic pressure. Is what that translates to. So I think that is very, very useful as like a biophysical model of the cell. And what they're really referencing is that the cytoskeleton, like the microtubule cytoskeleton, tubulin is incredibly negatively charged. So tubulin has got this folded domain with this negatively charged tail. And in fact, if we look at the charge of all tubulin family proteins compared to all unique proteins, tubulin has got like negative 20 or 30, which is much, much more negatively charged than the average proteins that they see. So they have these, long chains of carboxylic acids, which are cosmotropes. So the question is, where would ions go if they're just freely floating in the cell? And this is what Ling and these other people would say, that the ions should immobilize around the negative charges or hang out closer to the negative charges than just freely in solution. And when I say that ions are more attracted to immobilized charges, and what that means that if you have a polymerized microtubule versus just tubulin floating, over time, as this tubulin tumbles, this negative charge cloud moves. But if you're immobilized, it stays the same, right? And so what this means is that over time in the cell, if you're just hanging out, the potassium should then preferentially associate with the microtubule base structure, and it's almost like a capacitor, right? This is an electrical capacitor. You're building up a positive charge, and then you can do an action potential, and it releases the potassium somehow from that, and you get these cycles where you go back and forth between these states, and that's what they're measuring in yeast glycolysis, right? And there's some more interesting things about these negative charges, but I think I'm going to skip that in interest of time. I argue that this group observed these oscillations of ATP in yeast, but this group unknowingly perhaps also observed these oscillations of ATP in Drosophila melanogaster, in this coupling of activity, metabolism, and behavior across the Drosophila brain. They use a cool ATP sensor to measure oscillations of ATP as they stimulate the fly to move. So I think that this actually translates into higher organisms from yeast as well, is my proposition. So the summary of what I've shared with you today is that liquid-liquid phase separation and condensates organize biology, and that's really important, and I think there's strong data for that. water and polymers contribute, and I think that there's data to support that as well to contributions to liquid phase separation, and that to understand water's contribution, kind of understand water's behavior, and the most contemporary model is this rotational jump model and of water diffusion and how cosmotropes and chaotropes influence water affinity, which is important for understanding how these different chemicals interact with water differently. And then I take this to say that everything in biochemistry is precipitation reactions. You're either losing or gaining water, and that this is relevant also to formation of stress granules, liquid phase separation, and amyloid and protein precipitation. And this leads nicely into the biophysical model of the cell, where when you are now abusing the different properties of these cosmotropes and chaotropes, you get to organize the cell in these low entropy and high entropy resting and active states and cycle between them. So cycling between these two states becomes life. And I like this biophysical model of the cell because I feel it's kind of a bridge between fields. So biochemistry, cell biology, and genetics, they all kind of like operate in their own departments. But with this view in mind, then what they're really all doing is kind of measuring the water underneath, right? Water is what unites all these fields, and all these interactions can be explained in changes of water activity, potentially. So this is nice because then the biophysical cell model is kind of a bridge between all these different fields where water is the common language of biology. And so when you have chemistry and you have precipitation of salts, that's doing a counting of water. When you are folding a protein, you're doing the same thing where the protein side chains will be interacting based upon their relative water affinity. And that in cell biology, these yeast studies show that we can observe these cycles of potassium, ATP, water activity, and metabolites in oscillations in the cell. And this might provide lots of insight for how to understand how these systems are organized and how to help treat health and disease. And then when we're thinking of genetics and signaling, even when you're thinking of modifications like polyglutamylation or ribosylation, phosphorylation, acetylation, all of these are actually changing the properties of the chaotropes and cosmotropes, how these proteins interact with water and their water activity. And so again, all of these ideas can be kind of homogenized as changes in water activity. So then we can all be speaking the same language. So what are useful things to do in lab, like actual takeaway messages that might be helpful? Whether you choose sodium chloride or potassium chloride in a buffer, I would probably choose potassium chloride if the protein I'm working with is intracellular, or in the rare case it's not, sodium chloride. Crowders, I would probably do my biochemistry in the presence of Crowders for osmotic pressure. If cells are 300 mg per mil concentration, that's a lot of osmotically induced pressure to kind of influence them to behave and fold in ways where they might not in the test tube otherwise. And so when we're thinking of signaling, can also additionally think of as the signal is being transduced, how is it changing like the water polarization or ATP fluctuations? How is it affecting these great cycles in the cell? And then how does this change things like unique hydration environments as dictated by like the nuclear cytoplasmic ratio? And how does it change electrical activity or movement of ions inside the cells as well? And from that, I also want to say one thing about what have I told you that could be less useful. So all models are wrong, some are useful. What have I shared with you today that might be less useful, right? I've made a lot of conjecture, so where am I most likely wrong, or is there work to do? So the H2O theory of jump model is very contemporary and relatively accepted among chemists, but it's just a model based largely on computer simulation and interpretation. So some strains there. The two-state water model is also now gaining some popularity and is really useful, I think, to me to explain these models, but it's still not certain or proven. It is just a nice model to explain the anomalous behaviors of water, but I think a really convincing one to help understand life as well, and understanding how life might be using these two states of water for work. The law of water affinity that I shared is largely published in this 2019 paper by this one person, Dr. Collins. So there's not a lot, this isn't widely accepted or read. This is a very unique paper. So while I think it's a strong model, it has a lot to be left to be supported as well. In the case of water's involvement in liquid-liquid phase separation being important to understanding that phenomena, I think that many physical chemists are now trying to tell the biologists to pay attention to the water, and based on their models. And I think that there is relatively strong data that we should be paying attention. But I want to make sure that I'm not saying that stress granules have low density water or an outside is all high density water. I'm not trying to equate the granules to all low density water or high density water. Maybe there's a different ratio, or in general just a difference. I think the only thing I want to get away is that there is a difference inside and outside the granule and how the water behaves. But I'm not trying to say it's anything in particular. But when we're looking at phase separation, we're always looking at water and a protein, which is two components. So the holy grail of many chemists in this day and age is to try and find, or is to prove this idea that water itself as a single component system capable of liquid-liquid phase separation. And what that means is that you can supercool water below zero and not get it to freeze. And so you can do this in a fridge where you just have in your freezer frozen water, and then when you agitate it, it actually starts to freeze. So before the ice grows, it was super cooled, right? And so you can stably super cool water for a long time before it actually freezes. So as you have normal water and you're going down the temperature range and you're freezing it into super cold water, if you go down far enough, you eventually end up with high density or ices. But you can also approach this area that's called no man's land, where there's this theorized liquid-liquid limit, or liquid limit. So this is theorizing that water at the right temperature and conditions can spontaneously phase separate into the high density and low density phases of two-state water. And this is something that gets science articles occasionally, like this 2017 one, supercooled water reveals that secrets experiments provide evidence for two liquid phases in supercooled water droplets. Again, so it's almost like water itself being capable of doing this self-segregation under the right conditions. It's its own two-component system, where when you get to this theoretical point, you might be able to see a high density and a low density liquid. And again, people claim evidence of this, reversible structural transformations in supercooled liquid water, but these aren't perfect. This is still modeled. I think it's generally widely considered to be a good thing to pursue, but it hasn't been proven that water does this yet. But that also makes water extraordinarily unique in being a one-component system capable of liquid-liquid phase separation potentially. Then one thing I throw out, and this is mine, is that when we see liquid-liquid phase separation or granules form, that this is kind of causing overlapping hydration shells in the dense phase. So these things go from not touching each other to now their hydration shells have to interact with each other. And that's kind of like Collins' law of water affinity, where now you're under water activity or osmotic stress conditions where you're forcing this interaction and overlap. And so then I have these personal definitions where activity is the likelihood of making a new bond with water that I made up, but I think serves a useful tool. And same with osmotic pressure as a structuring of water, which of course may be a pseudoscience according to Wikipedia, but I think is also refuted by if you Wikipedia cosmotrope. Cosmotropes are defined as cosmotropic if they contribute to the stability and structure of water-water interactions. In contrast, chaotrope or disorder-making agents have the opposite, disrupting water structure. So we have to figure out if we're thinking of structured water as a pseudoscience or actually employing it in understanding cosmotropes and chaotropes and water's behavior. And in fact, the idea of cosmotropes and chaotropes aren't even settled. there's some people that write articles about this, like the water structure and chaotropicity, their uses, abuses, and biological implications. Some people say only chaotropes exist, things like that. And then the biophysical model of the cell is something that many, now a number of scientists use, and I think is really, really powerful, but this isn't widely accepted. And in fact, there's very little like literature on this, so it's a model that I think is really useful and could be really powerful, but there's a lot of work to be done there, and it's not certain about this. So some issues that are explicit is, I think the biophysical model of the cell, that Ling's work and the law of water affinity don't really match, because if we think that sodium has this like structured water around it, and the water in cells being kind of structured or high osmotic pressure, you expect these similar affinities, so you kind of expect sodium to actually diffuse into the cell, but it seems like that doesn't add up. So there's some issue with Ling's work with the law of matching water affinity that needs to be sorted out, and I think there's a few ways that could be done. And then when I talk about cancer being related to hydration, it's just not going to be so simple, but I think that there might be some overarching ideas there that are useful. So I've taken tons and tons of your time. I'm so thankful for you all letting me share this with you and record it so that I can use it later, and that's all I have. Thank you so much.